Alright, today we're going to look at our first lecture on biochemistry. This is one of your options for your IB chemistry test. Alright, so we're going to start out with more or less what is biochemistry? What exactly do we study in biochemistry? To make it simply biochemistry is the study of the chemistry of the human body or living organisms, just as it basically states in the name itself as it applies. We can see biochemistry. Bio for living things, chemistry, of course, you know what that is. Alright, so what else do we look at in biochemistry? Well, we study the molecules that make up living things, as well as the interactions of these molecules, and how we use food and convert it into energy, that energy that will power our cells in our body. And so biochemistry is a very complex subject. When you go to college, you're going to spend and take an entire course or several courses just on biochemistry alone. Okay, now all biological molecules in chemistry are organic. Organic molecules. Okay. Alright, now, in organic chemistry, there's some basic functional groups, just like the functional groups that we learned in organic chemistry. And this is a list of some of the common ones that we have that we will look at as we go through biochemistry. So, the first one that we are familiar with are like alcohols. And then we have carboxylic acids. The next one here is a new one. This is called a sulfhydryl group. And we'll talk more about sulfhydryl groups when we talk about proteins. And then this bottom one is a ketone, which you're familiar with. And then you have your good old aldehyde. And then this is an ester. And esters we'll find a lot in fatty acids that we'll look at later, and fats in general. And then you have your amine group, which are located in proteins, and then your phosphate group, which is kind of new for us, but a phosphate group is more or less found in nucleic acids, and so we'll only worry about phosphates for the HL and not the SL students. Alright. So next we have a list of all of the macromolecules that we're going to study in biochemistry. The macromolecules are basically the major molecule classifications that are found in living organisms. These are all gigantic molecules, reason why we call them macro, because they are so large. They are all polymers, and of course, let me erase that. They are all polymers, and remember that in organic chem, we studied polymers and how polymers are made, and we'll look a little bit more at polymers here as we go through this tutorial. Okay, now, the first classification of macromolecules that we're going to look at are proteins. Let's see, proteins. And then we're going to look at carbohydrates. And then lipids. And then for the HL students only, we're going to look at nucleic acids. Okay, so carbohydrates are simply like your sugars or complex sugars. Lipids are like fats and certain steroids. And then nucleic acids are DNA and RNA that many of you are familiar with. The functional groups for these different classifications will be discussed and addressed throughout this tutorial, specifically when we get into the individual macromolecules. And each classification of macromolecules will have its own tutorial lecture that will be posted here online. Now, as far as proteins go, the major functional groups that we see in proteins are the amine groups, the carboxylic acid groups. For carbohydrates, we often see ketones or aldehydes, one or the other, and then alcohols. They're all alcohols. Carbohydrates all have alcohol groups in them. And then lipids, commonly found ester groups and alcohol groups. And then there's some other groups that are in lipids too that we'll discuss later. And then nucleic acids, their big things are phosphate groups, but they also have alcohols because nucleic acids have some sugars in them, as you'll see when we discuss nucleic acids later on. Alright, so back to our idea of polymers. Remember, a polymer is a long molecule consisting of many similar building blocks, and these are called monomers. And monomers, as we discussed, are they're really small units, and then we link those units up over and over, and they're repeated. And those monomers eventually create the big gigantic macromolecules or polymers. There are specific monomers that make up each of the macromolecules. And for example, amino acids, which we have talked about in organic chem, and you're probably familiar with them in your biology courses, these are the monomers for proteins. So just to review, to go back and remember that. So say we have monomer A, and it's going to be linked to monomer A again, and another monomer A, and we just have this repeating chain of the same monomer over and over again. So in terms of proteins, this could be, each one of these could be amino acids that are linked together to create an overall protein structure. And we'll spend more time on that later. Okay, so this is basically how we link monomers together. These are known as condensation reactions. Write that up here. Let's see, condensation reactions. Okay, condensation reactions. And what we're basically doing is we're going to take one monomer that has an hydrogen, and we're going to link it together with another monomer that has an OH group. And when we link this together, we're going to lose water in this condensation reaction. So water is going to actually be a product in our condensation reaction. And so here we got our third monomer, and here's our fourth one right here. And we're just going to link those two together, and then now we are increasing our polymer chain by the loss of water. So condensation reactions, you always lose water in order to link monomers together. and this is a common theme throughout biochemistry. Okay, so the next slide here that we're going to look at is how we actually break the covalent bonds between monomers, and this is done through a process called hydrolysis. And hydrolysis is really just the opposite of condensation. Hydrolysis reactions bring in water to break the covalent bond, and that's our covalent bond right here. Okay, and it's broken. We break that by adding water, and then we now have our separated monomer. And then if we want to separate the 2 and the 3, we would actually just add more water, and we would break that bond through hydrolysis, and now we have another monomer, and we can keep on doing that, breaking down our macromolecular major polymer. Okay, so these two reactions, condensation and hydrolysis reactions, come very familiar with. They're very important. Okay, so now we're going to turn our attention to more or less a major theme in biochemistry, and that is energy. Energy is so critical for living organisms. We have to have energy or we cannot function correctly. We can't move our bodies, we can't think or do any of the normal everyday things that we need to do. Where do we get our energy from? Well, bottom line, it comes from food overall. Oops, let's go back. So it comes from our food. Food that we eat. Okay, now the sum of all of the chemical reactions that occur in cells in the body are referred to as your metabolism. And energy is made available in cells through a complex series of oxidation reduction reactions known as respiration. And most of you have taken biology and have studied cellular respiration in a little bit more detail. But just remember that all of the chemical reactions in your body are referred to as your metabolism, and respiration is the actual oxidation reduction process that is used, bodies go through to create energy. Now this process usually begins with a simple sugar molecule called glucose. And we'll study more about glucose later on. But glucose will be our starting molecule. It has a lot of energy in there. We're going to break those bonds through oxidation reduction processes, and through the respiration we are going to produce energy. Other energy-rich molecules are usually first converted into glucose or intermediates by metabolic processes. That just simply means that we can use proteins, nucleic acids such as DNA or RNA, and fats. And those fats can be basically used that sometimes we convert into glucose first, or molecules that are a little bit down the line, and then use those to create energy as well. Okay, so IB expects you to be able to calculate the energy that is found in foods. And basically how we do this in science, and you're very familiar with this already, is that you use calorimetry. Okay, now we use a special calorimeter. This is called a bond calorimeter. Oops. Erase that. Okay, a bomb calorimeter. And the idea is pretty basic, like most calorimeters. You're going to have an insulated container, so some kind of insulation. Okay, so that's insulated. Oops. Go back. Okay, we're going to have to start this again. So, okay, so here's our calorimeter again, our insulated container. Now this area is going to have a device we're going to put food in. This is where the food goes into. This right here is a container with water in it, H2O. It's going to be surrounding, and you're going to have a thermometer there. So this is for temperature, measuring temperature. And then what you're going to do is you're going to have a electrical current that runs through there. This is electrical heating cell, basically. And what that electrical cell does is it heats up the food inside the calorimeter, and so it starts the initial burning process. So the food begins to burn, and then it releases heat energy into the water, and we measure the temperature of the water, and then we can use our famous equation. The Q is equal to mass of the water times the specific heat of water times the change in temperature of the water, and this will allow us to actually calculate the amount of energy released by food. Okay, so let's actually do a problem here. So let's say we got a 0.7 8-gram sample of food. Okay, we place this food in a bomb calorimeter, and we have 100 point. No, let's actually go back and let's do 105.1 grams of water. That's the amount of water we're going to have in our calorimeter. And the temperature initially was 15.4 degrees C, and the change in temperature, or it's going to heat up, the water will heat up to 30.6 degrees Celsius. Okay? Now, what we're going to do is calculate Q of the water here. And if you remember, Q of the water then is going to equal the mass of the water, which is 105.10 grams. And we're going to multiply this by the specific heat of water, which is 4.18 joules. Gram degrees C. And we're going to multiply that by the change in temperature, which is 15.2. Maybe better erase this, make it smaller. Oops. Make it a little bit smaller here so we can fit it all on this. So 4.1 4.18 times the change in temperature, which is 15.2 degrees C. Okay, and now we got the change in temperature. Remember, we just take our final temperature and we're going to subtract it from our initial temperature. Okay, so that ends up getting us 677.63 joules of energy. Now what we want is units for this is kilojoules per gram of food. So we're going to divide this by our grams, our 0.6 grams or 0.78 grams of food, and this will get us a value of 856.1.1 joules per gram. And then we can further change that into kilojoules, which would just be 8.56 kilojoules per gram of food. Okay, so it's just a very simple calorimetry problem, just like what you've normally seen before. Alright, so that's our tutorial for introduction of biochemistry. Make sure if you need to go back and review this, take good notes on your note outlines, and if you have any questions, write down your questions, and then we can go over those questions in class and all of that. So anyway, until next time, have a good time.